Good evening, ladies and gentlemen. This is the last of a trilogy, I think. The first year I've been coaching cuttlefish, sepia officinalis. Last year I've been coaching jellyfish. And this year, the last of the trilogy, before I fade away into this. You always say, oh. Don't say R. We had a murder mystery last night. I'll see some of the cast here. And this is how we cultured this little sea urchin and why we do. Now, strangely enough, to culture these animals, you don't really need to know really in-depth biology of these creatures. The way it works for me is I've been breeding fish in the aquatic world for over 50 years. And someone would come along here, that's where the cuttlefish started. Someone would come along and say, have you ever bred cuttlefish? Ever bred sea urchin? No. So I would really look into the breeding strategy of this animal, of a animal. And then you take over, you become nature. And sometimes along that way you have to manipulate things. And you'll see how we manipulate things with the sea urchin. The animal itself is quite small. You'll find it all around our coast. And it feeds on seaweed, laminaria generally. It will feed on green seaweeds, but generally we feed them on laminaria. It also feeds on dry food pellets. The new year is a company. But how do we know that? Because at the moment, these are being farmed heavily in Scotland on salmon farms for food. And all that people eat is the gonads. It's not something I'm particularly like to eat the gonads of a sea urchin, but they do. And they are being farmed, this species is being farmed in Scotland big time. And they feed them, and the main places for culture at the moment for food is on the salmon farms. So what they found, if they put them under the salmon cages, they clean the salmon cages as well. And as they drop the salmon pellets down to feed the salmon, they found that the urchins will love and feed on salmon pellets. So when we're running a bit short on laminaria, we will feed them dry fish pellets, dry fish meat, singing pellets, and they will eat it, no problem. They also eat, and it's not here, but someone came to visit me and said, why don't you feed them cuttlebone? They love cuttlebone, and they get a stunning colour by feeding on cuttlebone. I said, you're joking. Well, we got loads of cuttlefish here. So I put in a full cuttlebone, and they cleared it. They chewed right through this cuttlebone, gone. And the colour on them really intensified, eating this cuttlebone. They're quite long-lived, up to 10 years. And there is a natural spawning period out here from about May to October. So generally, that's all I needed to know at the time. Why did we do it? Or why was I asked to do it? There was or is, a young woman, she's everyone's young to me. Very brainy, from Newcastle University, and she was working with our director, Colin Brownlee, at the time. And Colin Brownlee came to me and said, have you ever coached sea urchins? And I said, no. And he said, would you be willing to try? I said, yeah, we'll give it a go. So, I'm not going to say her surname, I can never say it. I know her Christian name, and she is lovely. She was really nice. And we got together, we had a meeting, and we discussed it, and we said, okay, we'll have a go. And I started reading up papers on how people actually culture these, the history of culture them. I'm always interested in the aquatic history of the animal culture. It's interesting to see how they tried it for a long, long time ago, back in Victorian times, right up through the present time. And it was incredible what she wanted to do for. She wanted the eggs. She wanted the gametes. And she is convinced she has found in these eggs of this little animal cells and genes which will break down cancer cells. And it makes, it's been science week this week, and we've had 3 days for the school children. And when I show this, the school kids, these are the urchins, and I'm telling them about them, and I say to them, reading this little thing makes it worthwhile to come to work. And it does. Because it's just for that alone. I can't tell you anything about her project. It's well above my head. But all she wanted was the eggs, and she wanted me to supply her with eggs. And this is the basis of her study. And she's doing quite well. So for my case, I started, as I said, always looking to the past to see what was happening in the past. And I found these old papers in our library. As you know, it's a fantastic library here. And I took up these papers, and I found that this guy at the top wrote this paper in 1847, and it was not a very good paper. It was described not with great accuracy, but they were having a go at culturing sea urchins. And then the next guy in 1969 was able to actually get them to spawn. And he set up this weird system. This was a black and white photograph of trying to keep the larvae alive. And up to this time, 1969, it was still very difficult to keep the larvae alive. And obviously since then they have developed it. And this is not my idea, this is the way it is done. Right. I say we, that's a lie. Pete collected the original animals. I think it was Exmouth. Yes, John, Exmouth. Yeah. Pete collected the original stock animals in Exmouth, and they brought them back, and then you have to sex them. And you get the males, you rattle the male. If he rattles, it's a male. The only way you can sex a sea urchin is under a microscope. And a lot of this work is under a microscope. So you look at the animal under a microscope, and if you look at the males, when we do so, next time, so we look at them on a microscope and we separate them, and we keep males in one tank, females in another tank, until we're ready for putting them together. You must keep them quite cool. If they go above a certain temperature, they will naturally shed their gametes anyway, like they would in the ocean. So you've got to be careful. And we keep the tanks on a light cycle, about 18 hours on a light cycle. You can see them greedily feeding on seaweed. They absolutely chunk this stuff. They gnaw into it with very sharp teeth. And then I say, to sex them, you've got to use a microscope. And you can't see it here, but when you look under the microscope, turn up the other way, you'll see the male has got a very short, distinct, conical genital patella. Very distinct. And the female is quite flat on the bottom. So you can set them quite easily under a quite low powered microscope. And then you separate them into their separate tanks, ready for breeding. And what we do, I work with Jolanka on this, both of us work together on this. And I'm also the animal welfare officer. Eve Southford came to me the other day. If you ever know Eve Southford, it's a lovely lady. And she was turning out in her office. And she said, "John, I think you'll be interested in this." And I reads the back of the file, and it said, "Shock inducer. Show to John Rundle." So I take it to my office, and I open it. 2 probes, and a bell battery, and a plug. You can see the road is, look, the plug is a round pin. And inside is a set of instructions. Type. 1954. An electrical method of determining the sex of sea urchins. You plug that in, you get your two probes separate like this, and this is battery. You get your quarters of sea urchin, and you if you shed gametes, it's a female. If you don't shed gametes, it's a male.
He's what it was.
I tried it out on Pete.
He's a female.
No, it's just the way he walks on me. That's what they used to do. It's incredible. This is the way we do it. We've already sexed them. They didn't obviously realise you could sex them by putting them in the microscope. So we sex them, put them in tanks. And then, what we do then is we use potassium chloride and we inject very carefully into the animal, male and female, 5 micromolar of potassium chloride. And then the eggs and sperm are collected in separate dishes and mixed together for fertilization. It sounds simple. Don't believe in me, it's not as simple as that. But that's the way it's done, and that's where you do it. So you have a set of beakers. I've also with my shock inducer, I've also thing. You have separate beakers. So you have a beaker with the males in and a beaker with the females in, and then a spare beaker with seed water. You inject them, both of them. Within seconds in breeding season, if you swirl the male around in the larger beaker with the seawater in, he will shed sperm. Put him back in the beaker. Get your female and swirl your female around in the jar with the sperm, and she will shed thousands and thousands of eggs. Then you get a twism stick, stir it around, voila. You got sea urchins. And then you have to have a special setup. We'll go on to actual development in a minute. Now, John Bishop very kindly allows me to use this room where he coaches his tiny obsidians. And John's got this ingenious stirrer set up in there, which I don't know who actually invented it, but it's been there for a long time. So you need these pieces of equipment. You need a controlled temperature room, which it is. You need filtered, very highly filtered seawater, very concentrated highly filtered seawater. You need a stirrer system, like you see, which we talk about now. You need a microscope all the time. This total microscope. And you need sieves for water changes, and you need a feed. You need an algae. Now, we are lucky in this building. We have one of the largest algae collections in the world, and I go to see my friend Richard Pipe, which I see often, and he lets me have this green algae, and we can feed. So I've got everything on to start production. What is amazing with some of the animals I breed is the size, the minute size. Some of it is incredible. It's not like cuttlefish, baby cuttlefish, 6 months long, you can see it and feed. Some of the smaller stuff we breed in culture is on the microscope, and it's amazing. See these things, both the metamorphs, the jellyfish, etc. This thing is absolutely incredible. It goes through these 6 stages until you get a spiky sea urchin. And you can, I won't read them out, but you can see it in the photographs more clearly. In your dish, in your bowl now, in that middle central beaker, you've got these eggs. And they're about 100 micromolar, very tiny, like dust. Within an hour, these eggs, they started to divide. It's incredible. All these photographs we took here in the microscopes, they started to divide. Within 12 hours, you got a mobile animal. The first stage of the urchin's life. You got this little creature. Like dust. I'm doing this under a microscope. Breeding sea urchins, you end up with eyes like, I'm not going to say it. You end up with big eyes involved. You end up with bulging eyes. You're always under a microscope. Because of the sheer size of the game. And then you start to go into the pretty stuff, the beautiful stuff. These things now are in tiny petri dishes right now, in John's room. In a little tiny petri dish, with a little stirring it around. Like the jellyfish, you remember last year I told you, when you're breeding, culturing these sort of things, you're here 7 days a week. You're here Saturday and Sundays. If you're doing it at Christmas, you're here Christmas Day. But we will do it Christmas now. It's the season. But you're here all the time. We are very lucky. We're never short of volunteer students. So we're able to, as a team, which can work continuously, looking after these other things. This is again microns. And this little thing is swimming through the water, just pulsating through the water in this tiny petri dish. A tiny dish. You have to change the water daily. Algae filled with water, you have to change the water daily. You've got to handle this thing. You're using very fine micro mesh sieves. And then you wash the sieves out with a water washer. It's time consuming. The water got to be changed. In 4 days you've got this beautiful little creature swimming around. In 10 days it starts to develop. And what you're looking at here, what you see on a microscope, it's incredible. This is not a sea urchin. Keep that in mind. It's not a sea urchin. Now it starts to get bigger and more beautiful. And again, it's micro. Your eyes are coming out your head. And it's pulsating through the water very slowly. Absolutely amazing. And you're still changing water daily. You're still sending these through micro sieves. Now, after 18 days, you start to see the sea urchin. Where's the sea urchin? Ah, not there. There's the sea urchin, there. That's the sea urchin. All this thing is a host of the urchin. That will die and fade away. Then what you start looking for daily, you start looking, there it is there. And then you start looking for a little arm moving, wiggling. This is pinhead. This is not very big. And you will see that arm there? Look? And once you see that arm moving, you know he's going to metamorphose and turn into a spiky urchin. So then you've got to be ready with these other dishes with little bits of laminaria and a tiny stone. Now I've had these stones out in the daylight in the seawater hole well before I started this project, and they have collected a biofilm on their algae. So you've got your dishes ready with your stone. And stirring around, this is stirring the water around. And this is ultra clean water. And when you see that little arm, you know he's going to turn into a spiky urchin. And you can watch it. You can watch it change in front of your eyes under your microscope, change into an urchin. Colin Brownie's always said he wants to film it, but we've never got around to it. And that thing, you'll see him, once he gets there, within hours, you can stay there and watch it, and you stay there in 2 to 3 hours, watching and looking and looking and looking. And you'll see that thing drop to the bottom of the dish now. And this case will drop away. And that little blob will become that. Your sea urchin. And this happens in the ocean. And it settles by that little tube front and come up, and in an hour, the larvae structure is collapsed, it's gone. And there in front of your eyes, it's this minute urchin on the bottom of the glass. And then you'll see it advance to the stone. It knows there's food there. It knows it can get a meal. And then you'll see it crawl across, and you'll see the one there and one there, and you'll see them crawl across, and you'll see them on the stones. And then we move them to bigger tanks, little dishes really. But again, every day you're changing water, changing water. And you're moving these things very gently. And you'll see them, you put more stones in, and you can see them quite plainly here. There's one there, there's another one there. And then they start to go on the stone, they start to feed. Once you know you've got that, you know you've got them. You know you've survived and you've got a collection of urchins. Our next stage, these are millimeter squares. And then we move them to small closed systems aquaria. We don't put them on the main big system out there. We set up proper closed system aquaria fish tanks and with special filtration, little tiny filtration systems. And then you can see, and the difference in size and growth rates is fantastic. They don't all grow at the same rate. These are the same animals that grow at the same time. You can see the growth rate. It's nearly a centimetre, and this one's still only a couple of millimetres. And they're feeding on, they're well crunching the laminaria. You can see them scouring through the laminaria. And then we've got them. You know we've got them. What we hope to do in the future, when Lambda comes back, we have collected, we have a collection of the big Aquarist, and we would love to get some eggs off these so she can look at the DNA in these eggs and see if they will help her in her medical projects. The ones we've got now are ones we've bred. She wants to come back and look at the ones we've bred and see if the DNA is different and then we'll breed those. She wants to see if the DNA is different in the cultured animals compared to the wild stock. This is all she moves when she comes back again. That's it, ladies and gentlemen. Thank you very much.